Good evening, students. Welcome to the channel, The Science Geek. Well, this is Ratna. I had made the last 3 videos which were from CBSE board. The lessons were from 10th standard biology and chemistry, acid, base and salt, and where I did for acids, then one I did for base and salt. Hope you all liked it as last minute revision. Then I started with control and coordination, brain, and I also did movements in plants, plant hormones, animal hormones, and a full recap of the lesson control and coordination CBSE. Well, life is a learning process, so let me explore the ICSE world. So just entering the ICSE world with 10th standard biology and a very interesting lesson, structure of chromosomes, cell cycle and cell division. Students, you all must have read this lesson from your junior classes because everything is interrelated, like what is a cell, the animal cell, the plant cell, the differences between them and the functions of the cell organelles found in them. For example, the first lesson of biology which is there in your 10th standard is all about the cell, animal cell and a plant cell, and you should quickly recap the differences which they have, right? Animal cell has a cell membrane, plant has a cell wall. The cell membrane does selective permeability, it is semi-permeable. The cell wall regulates the entry. The cell membrane is made up of lipoproteins. The cell wall is made up of cellulose. The cell membrane is living, it is flexible, and also it is living, flexible, lipoprotein, and the cell wall, especially plant cell, it is made up of cellulose and it gives the shape and rigidity. Then comes the nature of the cell organelles found, right? The other cell organelles mostly found in the animal cell are especially the nucleus and the role of the nucleus, which we will do in this lesson. In plants, exclusively the chloroplast, which will be done for photosynthesis. The plastids in plant cell, not found in the animal cell. Mitochondria, very much important cell organelle in the plant cell and the animal cell both. Then comes the other cell organelles like vacuole, a large vacuole in plant cell, small vacuole in animal cell, and lysosomes, and others which you did was endoplasmic reticulum, Golgi bodies. That was your first lesson, right? Now this lesson which we are doing will be the structure of chromosomes, cell cycle and cell division. Now what do you think, to which part of the cell organelle will it be related? It will be related to the most important cell organelle, nucleus. This nucleus is one of the most important cell organelle where it occupies in the central part of the cell. This nucleus, the features of the nucleus, if they ask you, I will give you a short revision or last minute revision for this lesson. The important keywords like nucleus. The moment you should say nucleus means what? The nuclear membrane, a round dense structure in it, the nucleolus. The cytoplasm within the nucleus is known as the nucleoplasm. And what does this nucleus contain? What does it contain? It is known as the brain house of the cell. Why? Because on this depends the entire entity of the cell. So this nucleus contains the fine thread-like structures which is termed as chromatin. This the cell will have when the cell is not dividing. Once the cell is ready to divide, entering the cell cycle, it has to perform the function of other things. Then what it will do? This thread-like structures will start condensing. It will start coiling and supercoiling like a telephone coil, and then they become distinctly visible under the microscope. At that time, these chromatin are termed as chromosomes. very well keep your ICSE book in front of you because your book is your Bible. I am just telling the key points of your book. So from page number 8, ICSE book, what are chromosomes? The nucleus contains the most of the cell's DNA, which is organized into discrete unit called chromosomes, right? Now these chromosomes, the structure was given by a German scientist, Mr. Fleming. He observed the larvae of salamander and he saw the cells were rapidly dividing. He termed it as mitosis. So Fleming, the names of the scientists are also asked. Who discovered what? So the first scientist here is Fleming. What did he discover? He saw the larvae of salamander and it's an amphibian, and he termed it as mitosis, a mode of cell division which you will study, and also he gave the thing of chromosome. It was studied by him only. Then comes chromatin chromosomes. Now what do these chromosomes carry? Brain of the cell. It carries the DNA, the genetic material. And this DNA is the genetic material of the cell, and a functional unit of DNA which becomes a unit of inheritance later on is termed as genes. So we started with nucleus, nuclear membrane, nucleolus, nucleoplasm, fine thread-like structures, chromatin. This chromatin, when the cell is about to divide, that's why when you perform the practicals, the nucleus takes the stain, because that stain which you put is the safranin stain, and with that stain you can see the mitotic mode of cell division in the onion tip cells, if your teachers can show you, and then you can see the chromosomes, and these chromosomes are made up of DNA, and DNA is nothing but the functional unit of DNA is known as the genes, because these genes are nothing but the proteins which we see in our body. This was the first part, right? Now, when I say you have to study the structure of chromosomes, means what points you should keep in mind, that what is the chromosome made up of, right? What is the chromosome made up of? So the chromosome is mainly made up of DNA, 60%, and proteins, that is 40%. So the nucleus is mainly made up of DNA, and this DNA is nothing, which is the percentage is 60% approximately, and the protein part is 40%. Now this DNA, another very important macromolecule of the cell. It was also, the molecular structure of DNA was given by Franklin, and later on in 1953 it's given in your book, and the two scientists who got the Nobel Prize for giving the double helical structure of DNA were Watson and Crick in the year 1962. On page number 9 is given. He will be prepared for your board exam. The name of the three scientists. Fleming, what did he discover? Then another one was Franklin. What did they discover? Then came Watson and Crick. What did they discover? Three scientists in terms. Okay, so this DNA, the chromosome is made up of DNA. Now this DNA is a very, very important macromolecule of the cell. This DNA is a double helical structure. It is having a double helical, a very complicated structure it has. For your 10th standard level, you must just know it is a double helical structure. This DNA is made up of the term, if you break the term DNA, there are two types of nucleic acid. It's a one type of nucleic acid, DNA, and another one is RNA. DNA and RNA. So this DNA and RNA are the two nucleic acids found in a cell. So DNA, deoxyribonucleic acid, RNA, ribonucleic acid, and they both are found in the nucleus. But we are now mainly concerned with DNA. Now DNA is again made up of, if you break the segment of DNA, a DNA is made up of a phosphate group, pentose sugar, and a base, and a nitrogenous base. This is a structure given in your textbook. I have highlighted this. You will also draw this for a board exam. If they ask you, show the double helical structure of DNA, what are the things it is made up of, and this whole thing, what it is termed as, this whole thing, the phosphate group, the sugar, the pentose sugar, and the base is called as a nucleotide. Nucleotide. I write the word nucleotide. What is a nucleotide is a very important question here. Okay, now The bases, as I told you, the bases are of 4 kinds, which is shown here: G and C, guanine, cytosine, thymine, adenine. So the 4 bases which are found in DNA are guanine, adenine, thymine and adenine. Guanine pairs with cytosine, thymine pairs with adenine. This pairing is done with the help of hydrogen bond. The only difference in RNA is having another base known as uracil. You can see on your page, DNA at a glance is given there. Kinds of nucleic acid, DNA and RNA. Components of the chromatin are DNA. Chromatin is made up of DNA, and the proteins are known as histone proteins. The components are nucleotide, which I just now told you, is a pentose sugar and a phosphate group and the nitrogen base. The nitrogen base are of 2 types, the purines and the pyrimidines. The purines are known as adenine, guanine, capital A, capital G. The pyrimidines are known as thymine, cytosine, uracil. This uracil is found in the RNA. The composition, nitrogen base composition RNA is adenine, guanine, cytosine, uracil. The nitrogen base composition in DNA is adenine, guanine, thymine and cytosine. If they ask you the difference between DNA and RNA, again an important question you can give in terms of the nitrogen base. And what are nitrogen base? Nitrogen bases are the components of a nucleotide. What is a nucleotide? A nucleotide is made up of a phosphate group, a pentose sugar and the nitrogen bases. And these all are the parts of a chromatin. Now, the basic structural functional unit of a nucleic acid, the basic functional structural unit of a nucleic acid is nothing but a nucleotide. As I told you, what is a nucleotide? This is one complete nucleotide. Phosphate, sugar, base. This is a nucleotide. And the hydrogen bonding is between the bases. So this is a nucleotide. Then comes, if they ask you the names of the scientists, so DNA at a glance, right? The structure, the nitrogen bases, and who discovered what. So the chromosomes, which are made up of DNA, this was the first thing which I wanted to tell you. So this is another thing which you should keep in mind. The nucleus is made up of chromosomes. The chromosomes are the long chains of DNA with proteins. This is nothing but the genes, and the genes are the specific sequence of nucleotides on the chromosome to encode a particular protein. So the genes are the unit of inheritance, as I have told you, the unit of inheritance. Also, another important term is there in your book, nucleosome. Nucleosome. Now this is there on the in context with, don't get confused with nucleotide. Nucleosome is when one DNA is associated with histone proteins. Usually one DNA is associated with 8 histone proteins. Like in a football, when you see the football, you see how all of them are, when you see the structure of the football from externally, how it looks like. So the DNA, the figure of that particular thing is given in figure 2.5 of your ICSE book, page 10. If you can see, always keep the book in front of you. They have shown the chromatid. The chromatid is supercoiled in the form of chromosomes because the size of the human nucleus is 6 micrometer. So the whole thing has to be fit into it. How will it fit into it? Unless it is supercoiled, it cannot fit into it. To study that structure only, this is the inner details of it. So that supercoiled structure is made up of DNA. DNA with histones, one coil is known as a nucleosome, and it is given here. The nucleosome becomes coiled, then supercoiled, and they get converted into chromosome. So the ultimate definition of chromosome, if it is asked to you, what is a chromosome? The chromosomes are highly coiled, ribbon-like structures formed by the condensation. The key word is condensation. Formed by the condensation of the chromatin fibers during cell division. This has to be written and underline the key words over here. I think this part is clear to you. Okay. Now we shift to what will this chromosome do? What do these structures do in the cell? What role do they do? After all, why it is required in a cell? They are required in the cell for what? Say if we eat food to get energy, and with that energy we work. So these things, and as I told, nucleus is the brain of the cell. So these things which are there, they will help the cell to evolve, to survive and make its own kind. That is, it will enter into a cell cycle. A cell cycle. A cell cycle means where the cell will divide, it will grow and again redivide. Grow, divide, grow and again redivide. This cell cycle, you can turn the page and come to page number 12, where the definition of cell cycle is given. The cell cycle is a series of events. Next question, important question here comes is, what is cell cycle? The cell cycle is a series of events that takes place in a cell, leading to the duplication of DNA and the subsequent division of the cell to produce the daughter cell. This is the complete definition of cell cycle. Now, how many stages it is made up of? The cell cycle is made up of 2 stages. First, the non-dividing phase known as the interphase. The non-dividing phase known as the interphase, also called as the resting phase. Next, a dividing phase known as the M phase or mitosis. Now, if they ask you, draw the cell cycle, just they ask you a question of what is a cell cycle, this definition. What are the 2 phases of cell cycle? 2 phases to told you, interphase and M phase. Now, this interphase is made up of 3 particular phases. First is the G1 phase, then is the S phase and the G2 phase. What is the G1 phase? The G1 phase is the first growth phase where maximum RNA and protein synthesis are there. The mitochondria and chloroplast, they divide, and that is the G1 phase. Okay. If they ask you what happens in the S phase, synthesis phase, here again more DNA is synthesized and the chromosomes duplicate but they remain attached. The chromosomes duplicate but they remain attached at a particular point. Now here I just want to tell you one thing. When you will see mitosis, you will see that a particular chromosome is divided into lengthwise. If you will start dividing it, this part is known as chromatid. Sister chromatid. One sister, second sister. The 2 sister chromatids are joined at the center by a particular point. That particular point is nothing but the centromere. The point of attachment of the chromatids, and with the help of this centromere, the chromatids divide. They go to the opposite poles. It is this centromere which helps the chromosome to align on the equatorial plate. So another important question, what is the centromere? Where it is found? What is its role? Okay, from chromosome only it is coming. Okay, so this is a very, very important lesson. So keep these small questions in mind. Previously, what was nucleotide, what is nucleosome, who gave the structure of DNA, what is chromosomes, then what is the cell cycle, what are the stages of cell cycle. Now comes what is centromere, where it is found, what is the function of centromere. So I was telling you the cell cycle is divided into these 3 stages, G1, S phase and G2 phase. G2 phase is the second growth phase where the more of RNA and protein synthesis takes place. Now, this mitosis, which you will study in bit detail as compared to meiosis, mitosis is divided into 2 stages. The first stage is karyokinesis, which is also known as the nuclear division, and the next one is cytokinesis, which is also known as where the cytoplasm divides to produce 2 daughter cells. And the sequence, you should learn the stages in sequence: prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. Okay, like this you should keep your fingers, your hands, everything ready to learn any subject. Just don't sit and read. You have to be very dynamic to learn or understand the concept of any subject. So I was telling you about a cell cycle. So now this was in this cell cycle told you the stages and what are the 3 stages, what it happens there, and what is the function of the cell cycle. The function of the cell cycle told you divide, grow and redivide. Like for example, your skin cells, the skin cells, they divide. You cut your nails, they again grow. Why? Cell division is there. Then the red blood cells, their life is only for 120 days, then again they are replaced. You see the box questions which are given here, all of them are important on page number 13. The bone cells, are replaced every 10 years. Then the epithelial cells are replaced after every 5 days. Then the liver cells after every 1 to 2 years. This is in case of animal. Now what about plants? Where does the cell division takes place in a plant? In animal cell, the cell division takes place throughout the body. But what about the plants, where it takes place? It takes place at localized region, in the meristematic part. The word meristem, if they ask you, now what are the kinds of meristem found in a plant? Apical meristem, lateral meristem, and intercalary meristem. Which part of the plant is called as meristem? The ever dividing. It is the primary tissue of the plant which keeps on dividing. And this meristem, if this meristem will not be there, the plant cannot survive. It is the first tissue formed in the plant. Then the secondary tissues like the phloem, the xylem, the parenchyma, sclerenchyma, etc. comes. So we are now concerned with cell division in plants, where it takes place in the meristematic tissue. What meristematic? It increases the length of the plant, and also for the gut, to increase the gut, cell division is required. So this is very important. What is the role of cell cycle and where does it take place? Okay, so this was about this part. In continuation of the previous part which we were doing, chromosomes, structure of chromosomes, DNA, structure of DNA, the role of DNA in the cell, the genes, and that was the previous part. So all these things are there in the cell. These are the raw materials which the cell requires to survive. That's why the nucleus is termed as the brain of the cell. Now if it is the brain of the cell, this brain, like your brain, what does your brain do? It thinks for you. It makes you do all kind of intellectual work, any kind of work, intellectual work, physical work, what you can say. Without brain, will you survive? You will not survive. So everything is related to your brain when we say that, and the nerve cell, which we say, the longest cell in the body, right? So like that, for the brain of in a plant or in an animal, what is their brain in a plant or in an animal? The nucleus. Now, why this nucleus is so important? Because of the constituents of which it is made up of. Now, as the name of the cell is, lesson is structure of chromosome, cell cycle and cell division. You did what is cell cycle, you read what is chromosome. Now we come to cell division. Now a cell has to divide in order to evolve. The evolution is nothing but when the cell gives rise to variation. Sometimes the cell may not give rise to variation, but that variation, and it is not necessary that mode of division where variation is not there, it is not important. It can be important. So for that only we say there are two kinds of reproduction in a organism: asexual reproduction and sexual reproduction. So this asexual reproduction is mainly related with one mode of cell division called as mitosis. So we will now go for mitosis. What are the stages of mitosis? How the mitosis goes in parallel with animal to plants? What is the significance of mitosis? Now, the mitosis is that mode of cell division where a single parent divides into two daughter cells. There is no reunion of two parents, single parent. So that's why I said asexual mode of division. Like amoeba, the yeast. Amoeba undergoes binary fission. Yeast shows budding. How? By this mitosis. So you can see on your page number 13, your book, that mitosis is divided into four stages: the prophase, the anaphase, the prophase, the metaphase, the anaphase, the telophase. These are the stages into which mitosis is divided into. Okay. So now, and the definition is, mitosis is that cell division in which one parent is involved and it produces two daughter cells. The number of chromosomes remain the same. Now please turn the page of your book and come to the diagrams which are given there. They are very important. Now what will you write for prophase? Interphase is the resting phase. Interphase is the resting phase where the cell will have the nuclear membrane, it will have the nucleolus, it will have the chromatin, the thin coil-like structures, and the cytoplasm is normal with centrosome. This centrosome we read in the previous lecture, cell organelle. What does the centrosome do? It gives rise to centrioles, and the centrioles give rise to spindle fibers. The spindle fibers, now you will see here what it happens. So we come to now prophase. What happens in prophase? Very nicely given in your book. The centrioles start moving apart. First point, the chromosomes become very distinct. They are divided, they become distinct into chromatids. Then the chromosomes get duplicated as chromatids. spindle fibers come, the centrioles are moving apart, and also the nuclear membrane and the nucleolus disappears, and this aster comes. What is this aster? Ray-like structures formed around the centriole. This dot is the centriole and the asters are ray-like structures formed around the centriole. Very important point, remember, asters are formed only in an animal cell. It is not formed in a plant cell. This is the first difference between the mitosis of animal and plant. So this word aster is very important. So this is prophase. So you can see this diagram there in your book also, and the points which are given here. After this prophase, which is the next stage, metaphase. What happens in metaphase? The chromosomes lie along the equatorial plane, along this plane, and each chromosome gets attached to the spindle, that is the spindle fiber, with the help of centromere. I had told you what is centromere. Centromere is the attaching point of two sister chromatids. The two sister chromatids, when they are attached, this is a cross. This point of attachment, this attachment is nothing but the centromere. Okay, I can show you again. This is this, this is this, this is this, this is this. So this point of attachment is nothing but the centromere. And this centromere plays a very important role. It helps the chromosome to align along the equatorial plate, and with this centromere, the sister chromatids, they divide in and go towards the opposite pole. So it is a point of attachment for the two sister chromatids. Centromere. Remember what is centromere. So here is the role metaphase. After metaphase, which stage? Anaphase. What happens in anaphase? See, in anaphase, the centromere attaching the chromatids start dividing. The centromere starts dividing here because here the sister chromatids go towards the opposite pole. The two sister chromatids are separated and are drawn to the two opposite poles by the shortening of the spindle fibers. Here the shape is different of spindle fiber, here the shape is different. You have to keep this in mind. And a small furrow appears in a cleavage type of thing, appears in the cell membrane here. This is anaphase. So that this diagram along the plate, here these two sister chromatids go towards the opposite pole and the spindle fibers, they shorten up, right? And the furrow appears here. Anaphase. After anaphase, which is the next stage? Telophase. What happens in telophase? Two sets of daughter chromosomes reach to the two set of poles. And again what happens here, please see your book, what's given here. Two sets of daughter chromosomes, that is chromatids, reach to the opposite poles. The spindle fibers again disappear because the cell has to come back to its original stage. The spindle fibers disappear, the chromatids thin out and they turn into chromatin fibers. The chromatids get converted into chromatin fiber, that is a thin-like structures as shown here. Okay, here the cell was dividing, here it is coming back to its original stage. And the nuclear membrane again is formed. This outer structure is the nuclear membrane. So these are the distinct points of telophase. If they ask you the differences between prophase, metaphase, anaphase, telophase, draw the diagram, write the points, underline the key points. Then, what is the next stage after telophase? Next stage after telophase, as I told you, mitosis is divided into two parts. Karyokinesis, this is all the nuclear division, and cytokinesis. Cytokinesis, the division of the cytoplasm. So, when this stage is reached, after this what happens? This cleavage deepens and it brings, and the cytokinesis means division of the cytoplasm. Another difference between plant and animal. Cytokinesis in animal cell takes place along the cell membrane, but in case of a plant cell, a cell plate is formed. You can see the book, it's given in your book. A cell plate is formed. Here, the cytokinesis, the complete cleavage is there on the cell membrane, and there a cell plate is formed. So I told you two differences here only. Aster in animal, no aster in plant cell. The cytokinesis in animal cell through a deep cleavage along the cell membrane, there it is a cell plate is formed and it goes inward. So this was the in brief about mitosis, right? Was in brief about mitosis. Now we come to what is the significance of mitosis. Now, significance of mitosis. This mitosis, as I told, differences you will keep in mind. Next question. The difference between animal cell and plant cell, told you, right? Asters are formed, no asters. I am repeating again. Cytokinesis by furrowing, in the cell membrane by cell plate. Occurs in most of the tissues throughout the body. Very important. Throughout the body it occurs, and in case of plant it occurs only in the meristematic part. Now, what is the significance of mitosis? This mitosis, it helps in growth, formation of new cells. It helps in repair, the repair of the wounded tissue. It helps in replacement of the old and the dead cells. It helps in asexual reproduction, where only one parent is involved, giving rise to two daughter cells. The number of chromosomes remain the same, amoeba and yeast. Amoeba by binary fission, yeast by budding, if you remember. And it maintains the same number of chromosomes. I think these are the points which will help you in revising the lesson cell structure and cell division for mitosis. Now, the next important part, and which you don't have to study in detail, is about meiosis. Meiosis. Another mode of cell division is meiosis. Now, this meiosis, also known as the reductional division, because this meiosis is so important that I am reading out that paragraph of your book, because the subject is learned by reading, writing and speaking. Meiosis refers to reductional division. It's a kind of cell division that produces the sex cells or gametes. So that is why it takes place in the testes and ovaries. Okay, it takes place in the reproductive organs, testes and ovaries in humans. And where it takes place in plants? In where? What are the sex organs in plants? The anther and the ovary. Anther produces the pollen grains and the ovary produces the ovules. If they ask you, where does meiosis takes place in a plant? Anther and ovary. And where it takes place in an animal cell? Testes and ovaries. What it produces? Testes produces sperm and the ovary produces the egg cell. Related to this, one diagram is given in your book where they have shown you the kind of cells like man and woman. Body cells, 46, total number of chromosomes which we have are 23 pairs, that is 46 in man and same 46 in woman. But what happens for the sex cells? The chromosome number they are reduced. Full is not taken here. That is what it is written in your book. The most significant aspect of meiosis is the number of chromosome in the sex cell is half. Half means here it is diploid. 46 is termed as diploid, 2N, and haploid is single N. So the sex cells, that is the sperm, will carry XY. If you go for the genetic constitution of the male chromosome XY, it carries 23 XY. The 23rd pair is the sex chromosome. In women it is XX. So the sperm and the egg, again when they will reunite during the process of fertilization, because this meiosis takes place in gametic cells. Like mitosis takes place in somatic cells and meiosis takes place in gametic cells, because it helps in evolution, it helps in fertilization, it helps in sexual reproduction, and it produces a new organism through fusion of gametes, through meiosis. And when the fusion takes place, fertilization takes place, the normal number of chromosomes is restored. That is diploid, 2N. So this whole thing of your book, the most significant part of meiosis is the number of chromosomes is reduced here. How it is reduced, I just now told you what is haploid and what is diploid. Now, the meiosis is divided into two stages. You don't have to read it in much detail. Meiosis 1 and meiosis 2. Meiosis 1 is known as the reductional division, and meiosis 2 is more or less like the mitotic division. Now the prophase of meiosis 1 is very important, and this prophase is divided, which you will read in higher classes, into 5 stages: leptotene, zygotene, pachytene, diplotene and dikinesis. This you will read, those who will take science in that 5 stages. And it is in this prophase that the important changes takes place. Now what are those distinct features of meiosis? What is the significance of meiosis? Significance of meiosis, please come to the last part of your lesson. Here the chromosome is halved, first significance. The chromosome number is halved, and that it happens in the gametic cells, and when it happens during fertilization. Now, second important thing here is meiosis provides recombination. It provides the genes to exchange of sister chromatids. Like in mitosis, the two sister chromatids were attached the two sister chromatids were attached. At the point of attachment was this centromere. But here the two sister chromatids will be from the chromatids coming from the two parents. They will exchange their arms. And this exchange of arms means genetic transfer, genetic changes, and this is known as crossing over. And the point of contact of the two chromatids is known as chiasma or chiasmata. There are two important terms are used here: crossing over and chiasmata. Crossing over and chiasmata. Very important, crossing over and chiasmata. It's given in your book, you see. The maternal and paternal chromosomes get mixed up during the first reduction division as they separate from the homologous pairs. Then, and this during separation only they get mixed up, and that time only crossing over takes place. And what is crossing over? The process of exchange of genetic material during meiosis between the two non-sister chromatids of a pair of homologous chromosomes is termed as crossing over. Figure is given in your book, figure 2.12. And this process results in the genetic recombination of offspring. Very important. And what is chiasmata? The point of contact. What is chiasmata? The point of attachment, as I showed you with the pen, between the two non-sister chromatids. The point of attachment, this point of attachment between this red pen and the green pen is known as the chiasmata. The point of attachment between the two non-sister chromatids of a pair of homologous chromosomes formed during meiosis is termed as chiasma, and it is a process which helps in crossing over. So this thing doesn't take place in mitosis. No chiasma formation, no crossing over. So now I think if you want to brief up the differences between mitosis and meiosis, your book has given you a very good table. First thing is mitosis in somatic cells, meiosis in gametic cells. Then mitosis helps in growth, repair and replacement. Meiosis helps in gamete formation. Second, mitosis takes place throughout the life, but meiosis takes place only in the reproductive cell. The mitosis helps in 2 daughter cell production, meiosis helps in 4 daughter cell production. Then the number of chromosomes in mitosis remains the same, here it is halved. Then another word which you should write is the diploid number of chromosomes are formed here, and here the haploid number of chromosomes are formed, but due to the fusion the normal 2n is again formed. Okay, then comes a single nuclear division is there, that is with the stages of mitosis, single one: prophase, metaphase, anaphase, telophase. But here 2 nuclear divisions are there, meiosis 1 and meiosis 2. Okay, and here the identity of chromosomes and genes in a daughter cell, identity of chromosomes and genes in a daughter cell remains the same, they are identical. But what about meiosis? They are randomly assorted. So I think a quick LMR of the whole lesson was what is nucleus, what are the parts of nucleus, and like nuclear membrane, nucleolus, nucleoplasm. What does the nucleus contain? It contains chromosome. What is the structure of chromosome? Chromosomes are the distinct supercoiled chromatin. What are they made up of? DNA and histones. Now what is the structure of DNA? Double helical structure. In this DNA, what is the nucleotide? The phosphate group, the pentose group and the base. How many kind of bases? Purines and pyrimidines. Purines, adenine, guanine. Pyrimidines, thymine, cytosine. Cytosine is replaced by uracil in RNA. A brief. After that comes what is a nucleosome? Nucleosome is a DNA with 8 histones. A nucleotide, what is a nucleotide? A phosphate group, a pentose sugar and a base is a nucleotide. This was DNA. Then comes the cell cycle. What is the cell cycle? 2 stages. Interphase and the M phase. Interphase is again interphase and the M phase. M phase is the mitosis phase. Interphase is G1, S and G2. That cycle which I told you. What is the function? Divide, redivide and grow. Cell cycle. Then cell division, 2 types: mitosis, meiosis. I hope I am clear with my lesson of chromosome, cell structure and cell division, and it was my first trial in ICSE for the students studying in 10th standard exam. So I think you will subscribe my channel, The Science Geek, and stay geeky and keep continuing reading, learning and be curious to learn more. And I think whatever I have told you, you have understood, and go through your textual exercises which is given in your book at the last part of the lesson, the multiple choice questions and the very short answers, the descriptive type, like whatever we did. And I think all the points I've covered, and I thank you. Hope to meet you again. Thank you and have a nice day.